Alright, good evening everyone, and thanks for sticking around for this last talk of the Faculty Bazaar. As you can see, I can't physically be here to tell you about our work tonight, so you're watching this video, which you can watch again on our lab's website, so don't worry about copying down all this on notes. And my lab is welcoming graduate rotators this year, as I minimize my talking head here. Our lab studies genome regulation by small RNAs in germ cells. Primarily, my lab is interested in the genetic phenomenon of RNA interference, or RNAi, which is an important process for regulating animal development. Many of you have heard about microRNAs and siRNAs that are bound by Argonaute proteins. My lab is interested in this third arm of the RNAi pathway called the Peewee pathway, which binds Peewee interacting RNAs. And this is a conserved pathway from flies to humans because it is essential for fertility. This pathway is expressed in germ cells in our gonads. Peewee interacting RNAs allow us to reproduce. But how are piRNAs made? This is actually a pretty poorly understood question. We do know that there are genomic regions called piRNA clusters which can give rise to primary piRNAs and ping-pong piRNAs. But there's a lot that we still don't understand. In addition, we do not fully understand how piRNAs regulate gene expression. We have 4 models for how they might work, but we would like to test these models in biochemical and genetic type of assays. One genetic approach is to try to knock out a piRNA cluster in an animal, and Jenny Klein in my lab is trying to do this in Xenopus using cutting-edge, pardon the pun, technology of CRISPR-Cas9. This CRISPR system is a nuclease that can cut a particular region in the Xenopus genome, and then we would like to mark that cut site, that mutation, with a transgene. And we had some success in getting this transgene in, but not quite what we would like at this piRNA cluster. So Jenny is trying to improve this transgene integration process by tweaking the CRISPR-Cas9 protein, putting on this extra module that allows us to tag on the transgene. A rotation project that we think can stem from this is to make use of this linker technology in Cas9 and then see if we can pull down chromatin, like piRNA cluster chromatin, that we can then do mass spectrometry on the proteins, like transcription factors, on piRNA cluster chromatin. Joe Clark is using Drosophila OSS cells, a tissue culture model that only makes primary piRNAs, to ask if we can dissect the mechanism of Peewee silencing using a reporter gene that he has been using a lot. And this reporter gene can be targeted by many piRNAs expressed in this cell line, and he has concluded that there is a bulk requirement of piRNAs, primary piRNAs, that is needed in order to trigger gene silencing. And this work is in review at RNA. A rotation project stemming from this is that we would like to develop a second insect cell line from mosquitoes that expresses ping-pong piRNAs. This is different from the primary piRNAs in these Drosophila OSS cells. We now have this AAG2 cell line that we have confirmed expresses a variety of Argonaute and Peewee proteins and make robust piRNAs. So in this rotation project, we would like to make reporter genes that can be targeted by mosquito piRNAs, and we have evidence that transfection in these mosquito cells works pretty well as Drosophila S2 cells. Yulia Sitnikova in my lab is also making use of these Drosophila OSS cells, and she has shown that Peewee proteins, in addition to binding piRNAs, can also bind long non-coding RNAs, as shown in these genome browser views. These are 87 and 6 kilobase windows showing these extensively long non-coding RNAs being bound by Peewee. And interestingly, these long non-coding RNAs are coming from de novo transposon insertions that you normally would not see in the genome. She has characterized another variant of this OSS cell line called OSC cells and showed that these cells have very different dynamic transposon landscapes. Using a genomic PCR assay she has developed, she has shown that these OSC cells have different transposon landscapes from OSS cells and vice versa. In fact, you can see here by this diversity of transposons that the classes of transposons are quite different in their proportion between OSC cells and OSS cells. And this work is in review at Genome Research. A lot of the work we do could not be done without the help of Riazur Rahman, who is a joint postdoc with my lab and Michael Rosbach's lab. He helps us in bioinformatically process the huge amounts of data we get from doing deep sequencing on the Illumina Deep Sequencer here at Brandeis. And with his code, we've been able to discover these de novo transposon insertions. He's also developed a program that we are tentatively calling TIADA, which can also detect transposon insertions and deletions in fly lines that have resequenced the genomes amongst these inbred fly lines. We've compared our program TIADA to another program called TEMP and find that our program TIADA can detect in blue many new transposon insertions missed by the other program, although we have pretty good overlap in this red region as well. More recently, we can also detect transposon deletions. And a third rotation idea would be to see how our program can determine transposon insertions and deletions in a variety of other Drosophila cell lines that are very important for biochemistry, and these genomes have been completely sequenced. They're just waiting for us to analyze and then confirm by genomic PCR. We would also like to check in those fly lines, those DGRP flies, whether the insertions and deletions that TIADA discovers are true or false, and which program is better, our program or the competition out there. If you rotate in my lab, you can help us figure this out. Finally, since there are a lot of neurobiology students out there, I'd like to also throw this out as a fourth rotation project idea. Michael Rosbach, in collaboration with Scott Waddell's lab, had published in Science that Drosophila fly brains may also have very dynamic transposon landscapes. And we have shown by Western that there's a small amount of peewee in fly brains, and so we're intrigued by this idea that transposon landscapes might be different between the fly brain and the ovary here. So if you're interested in this, we can make some genomic DNA and sequence and determine transposon landscapes between the fly brain and the fly ovary. So, as I mentioned again, my lab is open to rotators. You can visit our lab here in the Rosenstiel building. You can see my lab here, getting together, doing things. And you can view this video again on the Lau Lab website. Thanks very much. I can't take questions, but I look forward to talking to you later in the year. Thanks.